Hello student and welcome to my class. Today we are going to study on the topic restriction endonuclease. So let us move ahead and see what is restriction endonuclease all about. So restriction endonuclease are also known as restriction enzymes, and they were discovered by Werner Arber, Daniel Nathans and Hamilton Smith. For this, they received the Nobel Prize in the year 1978 in medicine. So basically, these restriction endonucleases are also known as restriction enzymes. So in short, let me write here, they will be known as RE. Okay. Now, these restriction endonuclease is a protein nuclease, and this nuclease recognizes a specific short nucleotide sequence of DNA, which means that until and unless they recognizes the site which are known as a restriction site or the target site for the restriction enzyme, they are not going to perform their function effectively. So a recognition of the site is very, very important. Now, and then what will they do once they recognizes the site? Once they have identified their target sequence, they will then cut the DNA only at that specific site. So this line only at that specific site will matter depending on the types of restriction endonucleases. But for now, we will be saying that these restriction enzyme identify the sequence. Once they identify the sequence, then they will cut the DNA sequence. Okay, I hope it is clear till there. Moving ahead, the restriction enzyme are found in bacteria, or in other words, we will say prokaryotes. Okay, and each restriction enzyme recognizes just one or a few restriction sites. So every restriction enzyme that are found in these prokaryotes have their own restriction site. Okay, they identify a particular site in the DNA, and based on that, they will be cutting the DNA. So they are not going to cut randomly. Okay, so after recognizing its target sequence, a restriction enzyme makes a double-stranded cut in the DNA molecule. So we all know the structure of DNA molecule, which is a double helix. And this double helix, once the recognition site is being identified by the restriction enzyme, they are going to cut not only in the single strand, that is 5' to 3', but they are also going to cut from the 3' to 5'. Okay. Now, typically, the cut is at or near the restriction site. Just like the first definition I've told you, they are going to cut at the specific site. But again, depending on which restriction enzyme we are talking about, some restriction enzyme cut at the specific site they recognize, but some restriction enzyme cut a bit away from the recognition site. Okay, but the pattern is predictable. Okay, moving on. So this picture depicts a very clear, idea about what a restriction enzyme does or what it do, right? In what is its function in the system. So you can see this is a restriction enzyme which have 4 to 6 base pair long. These restriction sites are of 4 to 6 base pair long, and once the restriction enzyme, which is represented by the scissor, identify these sites, then they are going to cut it. So in short, we will also call the RE or the restriction enzyme as molecular scissor. So these molecular scissors are going to cut at the sequence of the DNA that they identify. I hope it is clear. Okay, so moving on. Now, more than 400 restriction enzymes have been isolated from bacteria and that manufactures them. Okay, so you will have different uh, restriction enzymes being synthesized or being manufactured from different prokaryotes or different bacteria in short. So in live bacteria, restriction enzyme function to defend the cell against invading viral bacteriophages. So let me just emphasize this line more. Now we are saying that these restriction enzyme are going to cut the DNA sequence, right? They will identify the DNA sequence. Once they identify the DNA sequence, then they are going to cut it. Now, we are also saying that these restriction enzyme act like a defense mechanism or like the warriors. Like say, for example, if any viral or bacteriophage will enter inside the bacteria cell, these restriction enzyme are like the soldiers of the bacteria cell. They are going to chop this bacteriophage, the DNA of the bacteriophage that is injected inside the bacteria. But at the same time, even the bacteria will have its own DNA. Okay, so we have to keep into consideration, or this question should come in your mind that will not the restriction enzyme then cut the DNA of the bacteria also? Because if we are talking about just 4 to 6 base pair long recognition site, so that same recognition site may be present in the DNA of a bacteria also, right? But we will study ahead how does these restriction enzyme identify the difference between the difference between the sequence of the bacterial DNA as well as the uh, viral bacteriophages that has invaded a bacteria. Okay, so restriction enzyme of the bacteria, it cleaves the recognition site in the viral genome. So again, these restriction enzyme identify the recognition site. Once it identified the recognition site in the viral genome, then what will they do? Then they are going to fragment and destroy the DNA of the invading bacteriophages. So basically, the bacteria does not like anything that is extra. Okay, any one of us does not like anything that comes extra, that was never present earlier, and most important, that can be harmful to the cell or to the bacteria itself. So here these invading bacteriophages are going to inject their DNA. Once they inject the DNA, they will want to incorporate the DNA into the bacterial genome, and that is what the bacteria do not want. So basically, these restriction enzymes are present in prokaryotes to act as the warrior or the soldiers or the fighters that are going to destroy the viral DNA or the bacteriophages or any unwanted extracellular DNA that it is entering or that wants to enter inside the bacterial cell. I hope it is very clear. Okay. Now, so if you look at this picture, you can see a very, I mean, you will understand it even more clearer that if this is a bacteria and you can see this bacteriophage which has infected a bacteria and it is injecting its genome or DNA inside the bacterial cell, then what is going to happen? The circular pink colored dot represent the restriction endonucleases. Immediately, these restriction endonucleases are going to bound to these viral genome. Okay. And once they bind to the viral genome, what they are going to do? They are going to cleave and make them inactivated. So now these viral genome, even though they have entered into the bacterial cell, but they are incapable now or inactive to harm the bacterial genome. Clear? So just like I've told in the previous slide that if these restriction enzyme are identifying, okay, they are identifying a specific sequence in the viral genome and then cutting them and fragmenting them, so will they not identify the same sequence that will be present in the bacterial genome? Definitely they are going to, right? Because we know that the sequence which is just 4 to 6 base pair are going to be similar or they will be present in a bacterial genome also. So how will these restriction enzyme identify or differentiate? So in this second picture, if you see, you can see this ME. This ME stands for methylation or methylated region. So what will happen is the bacterial genome will then be methylated by the one of the subunit of the restriction enzyme itself. So they are going to tell the bacterial genome that, see, I don't want to degrade you because I am part of you. So please protect yourself. So how do you protect yourself? You protect yourself by being methylated. And once you are methylated, I am going to identify that you are mine because you are methylated, and any DNA that is not methylated is the one that I have to target and I have to destroy and chop them into smaller pieces. Is it clear? So I think it is very clear that how the restriction enzyme differentiate between the extracellular and the bacterial genome. So one is methylated, the other is not methylated. So interestingly, a bacterium is immune to its own restriction enzyme even if it has a target sequences which could be destroyed by its own restriction enzyme. So basically, the point is, if the bacterial genome is not methylated, then the restriction enzyme is going to fragment it, is going to cleave it and make it into smaller pieces or destroy it completely. But why it is incapable of doing that? It is because these bacterial genome are highly methylated when any extracellular DNA enter inside the bacterial cell. Okay. So here you can see how a methylase enzyme adds a methyl group. And when I say methyl group, it is represented by CH3. Where does it add? It adds to the adenine or to the cytosine bases. So wherever it's CA or it's CC, okay, in the recognition sequence, the methylase enzyme is going to add the methyl group, and in the process, it will modify and protect from its own endonucleases of the bacterium, and the restriction enzyme and its corresponding methylase constitute the restriction modification system of a bacterial species. So I think it is very, very clear from this definition that the methylase is going to add a methyl group in the bacterial genome, and once it's add the methyl group in bacterial genome, the restriction enzyme, even though it recognizes the sequence, but it is not going to be able, okay, it cannot be effective cutting of the bacterial genome. Why? Because it has been modified. Okay, so methylation is a very important step in the process of when the bacterial cell is attacking or it is destroying the extracellular DNA. Okay, so here you can see this picture is a very clear representation of how methylation is happening. So methylation is happening at the restriction site of a bacterial genome. You can see the C represented by green color and M represented by purple color. So wherever there is a C, there is also an M, and wherever there is an M, there is a protection of the bacterial genome. Clear? And if you look at the cytosine without methyl group is different, and the cytosine with a methyl group is different. So once it is different, which means there will be conformational changes, and if there is going to be a conformational changes, it will not be identified by the restriction endonuclease. Okay, and as you can see, who donates this methyl group? It is the S-adenosyl methionine, which transfer, okay, or which is a donor of the methyl group. So from SAM, it gets converted to SAH, that is S-adenosyl homocysteine. Okay, I think this picture is very, very clear. This figure is very clear and it's telling us a clear picture of how methylation is taking place. Next, Now, there is also a nature of cleavage by restriction endonucleases. Okay, the nature of cleavage produced by a restriction endonuclease is of considerable importance. Now, what is the nature of this restriction enzyme? What type of fragment are they going to produce? All these things are very, very important, and we have to take them into consideration for many other purposes or application of these restriction enzyme. So they say that they cut the DNA molecule in two ways. First, many restriction endonucleases cleave both strands of a DNA simply at the same point within the recognition site. See, within the recognition sequence. Wherever it is identifying the recognition sequence, it is cutting at the same point, simply cutting at the same point. Okay, so as a result of this type of cleavage, the DNA fragment with blunt ends are being generated. So why? Because they are cutting exactly where they are recognizing them. So some of the example like PVU2, HAE3, ALU1 are the example of restriction endonucleases producing blunt ends. And blunt end may also be referred to as the protruding ends. Okay. So some of the examples, see, this figure, if you look at the right hand side of the figure, that is a blunt end restriction endonuclease. So how is this a blunt end? Because see, wherever there is a recognition site, there exactly at that place it is going to cut, and when it cut it produce no overhangs, or there is a uniform edges, and when there is a uniform edges, this kind of fragment is known as a blunt end. Okay, on the other hand, a style of a cleavage by the restriction endonucleases, the two strands of DNA are cut at two different points, and such cuts are termed as staggered cuts, and this result into a generation of protruding ends, that is one strand of the double helix extend a few bases beyond the other strand, and such ends are called cohesive or sticky end. I think this picture depict it very, very beautifully. Let me just delete this line. Okay. So if you look at this figure, you will see what is the difference between a sticky end and a blunt end. So when we are saying a sticky end, see, when a restriction enzyme recognizes a recognition site, it is going to cut the DNA strand, but it is also going to produce complementary overhangs on both the strand. Clear? Unlike if you are looking at the blunt end. So when the blunt end are produced, the cut is exactly at the same side for both the strand. Clear? Which is different in a sticky end. I hope it is very clear. Okay. Okay, this is a very good example when we want to see the types of DNA cut generated by restriction enzyme, EcoR1, and the other one is EcoR5. So EcoR1 is producing sticky end cutters, whereas EcoR5 is going to produce the blunt end cutters. So here, see, if you look at EcoR1, the recognition site of EcoR1 is GAATTC, but it is going to cut between G and C. Similarly, it will be on the reverse strand or on the other side of the strand, GAATTC. So it is going to cut at between G and A, but from 5 prime to 3 prime strand. Okay. And after the cut has taken place, you will see the sticky ends that are being produced by these EcoR1 enzyme or such similar enzyme that produces the sticky end. Whereas if you look at the blunt end, here the example of EcoR5, the same sequence is being given to EcoR5. It recognizes the same target sequence, but the cut is at a different position. So the cut is between TA and AT on both the strand, and you can see there is no overhangs of the strand of the fragment, and it is completely blunt. So this is a very good example between a sticky end cutter and a blunt end cutter. So basically, restriction enzyme are of two types. Either they produce sticky ends or they produce blunt end. Then if you look here, there is a following table that shows sticky and blunt end generated in the restriction site by various restriction enzyme. So EcoR1, the source of EcoR1 is from Escherichia coli. It produces sticky N, that is the recognition site given to you in the third column, and the type of cut it produces, it will produce sticky N. Whereas BAMH1, which is isolated from Bacillus amyloliquefacin, this third columns identify or signify the recognition sequence, and this will also produce sticky, and similarly will be for TAC1, Thermus aquaticus. The recognition site, it is a 4-base pair, and it also produces a sticky N. Whereas the last one, that is ALU1 or ALU1, it is isolated from Arthobacter luteus. Its identify recognition site is also of 4-base pair, and it produces blunt N. Okay. So here with a very good example you can see as an example of how a restriction enzyme recognizes and cut at a DNA sequence. So let us consider EcoR1. If we consider EcoR1 as an example, a common restriction enzyme used in biotechnological lab, the EcoR1 cut at the following restriction site. So if you look at the sequence GAATTC, which is in the strand of 5' to 3', in the reverse strand, you can see it is CTTAAG from 3' to 5' strand. So this is basically the one highlighted in pink. It is the EcoR1 site. Example, if we place the two DNA strands together in a straight line, they appear as palindrome. Okay, so what is palindrome? Palindrome sequences are mirrors images of one another. So if you check here, 5' to 3', GAATTC, whereas 3' to 5', it is CTTAAA. It's like when you look at yourself in the mirror, you see your right to be the left and left to be the right. You are just the mirror image or the opposite of one another. Similarly is the case of these palindrome sequences. So whenever we talk about palindrome sequences, I hope you'll remember from now onwards that they are the mirror images of one another. So a palindromic sequence is a nucleic acid sequence on a double-stranded DNA or RNA. Now, wherein reading 5' to 3' N forward on one strand matches the sequence reading 3' to 5' N on the complementary strand, with which it forms a double helix. So the top strand read GAATTC, while the bottom strand will read CTTAAG. So if the DNA strand is flipped over, the sequences are exactly the same. So I hope it is very clear when from now onwards if we use the word palindromic sequence, it will be very clear, and remember that they are just mirror images of one another. Okay. Now, if you look at an EcoR1 enzyme that binds to the EcoR1 site, it is a piece of DNA and makes a cut on both the strand of the DNA, right? As shown below, if you look here, 5' to 3' strand is GAATTC, whereas the 3' to 5' strand will be CTTAAG. But the cut site is same in both the strands. It is between guanine and adenine. So when EcoR1 recognizes and cuts its recognition site, it always does so in a very specific pattern that produces N with single-stranded DNA overhangs. Thus, it produces an overhangs of 5', that is AATT, okay, on both the strand. Okay, moving ahead. So this is the pattern. If you look at this pattern, it is very, very clear how EcoR1 enzyme is going to cut both the strand of a DNA. Once it cut the strand of the DNA, where the cleavage is happening or where the cut is happening, and you can see these overhangs that are being produced by the EcoR1 because it produces the sticky end. I hope it is clear. Okay. So, the two sticky ends may join together by a complementary base pairing. Now, if another piece of DNA or a foreign DNA has matching overhangs, it can occur when both DNAs are cut with the same restriction enzyme, such as EcoR1. And the overhangs of the two different DNA stretches can also stick together by complementary base pairing. So, if you look at this diagram, the last one, you can see that we have DNA 1 and we also have DNA 2. And if I'm cutting DNA 1 with EcoR1, I'm also cutting DNA 2 with EcoR1, then we will have a complementary uh, overhangs. Okay, and these complementary overhangs can then form a bond or can then be joined together and complete the DNA sequence. Okay, it will complete the strand. There will not be any fragment or there will not be any cut. So this is how the concept of gene cloning have started. That you cut the two different DNA but with the same enzyme. So when we cut the different DNA with the same enzyme, we are going to get the same complementary overhangs and then we can join them. Okay. So for this reason, enzymes that leave single-stranded overhangs are said to produce highly reactive sticky end on the broken ends. Okay, so if you have a single-stranded overhangs, like both the complementary have the overhangs, so the presence of these overhangs in the strand make them very, very reactive. So they are always ready to form a bond with the any complementary sequence they come across. So sticky ends are helpful in cloning and genetic engineering. So this is one of the reason why the discovery of restriction endonuclease was very important and was it was a great revolutionary in molecular biology because it helped cloning and genetic engineering because they hold two pieces of DNA together so they can be linked by DNA ligase enzyme. Okay, so these restriction enzyme are molecular scissor, whereas DNA ligase are molecular glue. So they will join them together. Okay, moving ahead. Now, not all restriction enzyme produce sticky ends. So now when I say that sticky ends are very important because they are going to help us in cloning and genetic engineering, but we also have to keep in mind that not all restriction enzyme produces sticky ends. So there are some restriction enzyme that produces blunt end, or they are known as blunt cutters, and they are going to cut straight down the middle of a target sequence and leave no overhang. So for example, we have SMA1 enzyme. This SMA1 enzyme is going to cut exactly in the middle of the recognition site and produces blunt end. Okay, so a SMA enzyme binds to the SMA restriction site, which is given as follows. So you can see the recognition site for SMA1 is CCCGGG, and 3' to 5' it read as GGGCCC. So it makes a cut right in the middle of the sequence on both the strand and then produces blunt end. So you can see the cut site is between cytosine and guanine, and even in the other strand it is guanine and cytosine, so it cut exactly in the middle. There is no overhangs being produced by this enzyme. So blunt ended DNA fragment can also be joined together by DNA ligase, just like I've said in the previous slide. Now, however, blunt-ended fragments are harder to ligate, okay, or harder to join together during the ligation reaction. Why? Because it is completely blunt. There is no overhangs. Unlike the overhangs of fragment, what are they going to do? They are always reactive because they want to form a bond with someone, some other strand. So once they identify the complementary strand, they will immediately form a bond between the two strands. So this is a drawback, so to say, in a blunt fragment where such kind of joining or such kind of ligation will be difficult to happen. Okay, I'm not saying it is not going to happen, but it is going to be comparatively less or more difficult compared to the sticky end. Okay. So here, if you look how what we will take the class of DNA ligase in the next class, but in the next topic. But now you can see here that what DNA ligase basically will do. So DNA ligase is a molecular glue or a gum which will join together the cut end of DNA or the sticky end or the blunt end by creating a phosphodiester bond necessary to complete the sugar phosphate backbone of the newly synthesized transgenic DNA. So here you can see this circle blue color is the phosphodiester bond, and this phosphodiester bond is created even though if there is a fragment by DNA ligase. So this topic we will discuss in the next session or in the next class. Now, basically, there are also types of restriction enzymes. So, when I say if you remember me repeating the line in the first slide that when we said that restriction enzyme are going to cut exactly where they identify their recognition site. But again, that will depend from enzyme to enzyme. So here in this topic, we are going to see that there are three main type of restriction endonucleases. We have the type 1, the type 2, and the type 3. So let us see each type and based on that do put more emphasis on what are the main difference between these three types of restriction enzyme. So these are the complex type of endonucleases which cleave only one strand of the DNA. So not going to cut every time both the strand, but here this type 1 RE is going to fragment or going to cleave only one strand of DNA. And these enzymes have the recognition sequence of about 15 base pair in length. So the sequence also, if you remember the first figure, we wrote 4 to 6 base pair. But here, sometimes the recognition sequence also can vary up to 15 base pair in length. They require magnesium ions and ATP for their functioning. So in the absence of magnesium and ATP, this enzyme cannot perform effectively or cannot perform at all. Now, such types of restriction endonucleases would cleave the DNA about 1,000 base pair away from the 5' end of the sequence TCA located within the recognition site. So what will happen is these type 1 RE or these type 1 restriction endonucleases will identify the recognition site. Once they identify the recognition site, the cut will not happen there. The cut is going to happen 1,000 base pair away. Clear? So type 1 identify 1,000 base pair away or it will cut 1,000 base pair away from the recognition site. Some of the important example of type 1 restriction endonuclease are ECO-K and ECO-B. Okay? I hope it is clear. So let us move on to type 2 restriction endonucleases. So whenever we say that, restriction endonuclease was a great revolution or a great discovery where molecular biology was taken up to a higher height, it is because of this type 2 restriction enzyme. Because these type 2 restriction endonucleases are the important out of the type 1, type 2, and type 3 are the important one because they are the one that are most suitable or only suitable for gene cloning. Okay, so let us see. These are the most important endonucleases for gene cloning and hence for recombinant DNA technology. Uh, just to remind you that recombinant DNA technology means if you have an extra DNA that belongs to maybe a different plant or a different species, but you want to incorporate that gene into this uh, already existing DNA, okay, then you will produce the genome that is an addition of this plant B. Okay, so for example, you have plant A and plant B, and you want to take the gene from plant B and incorporate into the genome of plant A, then that process, okay, when your gene of plant B is incorporated into the genome of plant A, that DNA now will not only be known as the DNA of that particular species, but it will be known as a recombinant. Why? Because you have recombined a few segment of the gene into the existing one. I hope it is clear. So these enzymes are the most stable of type 1, type 2, type 3. The type 2 are the most stable one. They show cleavage only at a specific site. So once these restriction endonucleases have identified the target sequence of have identified the recognition site, they are going to cut exactly at that point. And therefore, they produce the DNA fragment of a defined length. So there is no variation of length, but you will know that if this particular enzyme is going to cut the genome, what will be the length produced by these enzymes. These enzymes show cleavage in both the strands of the DNA immediately outside the recognition sequences. So basically, unlike type 1, where it is going to cleave to only one single stranded DNA, here double stranded DNA is going to be cleaved. They require of magnesium ion for their functioning. So if you remember type 1 requires ATP as well as magnesium, whereas in type 2, we requires only the magnesium for its proper functioning. And such enzymes are advantageous because they do not require ATP for cleavage and they cause cleavage in both the strands of DNA. So, at a very less or no energy requirement restriction enzyme, we are going to not only get cut a single stranded DNA, but a double stranded DNA is going to be cut here. Okay, so because when we say it requires ATP, ATP means it requires energy. Okay, so only type 2 restriction endonucleases are used for gene cloning due to their suitability. So again, the recognition sequence for type 2 restriction endonuclease enzyme are in the form of a palindromic sequences with rotational symmetry. So unlike type 1 and type 3, type 2 is going to recognizes the sequence site. Once it recognizes the target site, it is going to be a palindromic sequences, which means all type 2 restriction endonucleases have a restriction site of the palindromic pattern. Okay. Okay. So that is the base sequence in the first half of one strand of DNA is the mirror image of the second half of the other strand of that double DNA double helix. Important example of type 2 restriction enzyme or restriction endonucleases will include NIF-F1, ECO-R1, PVU2, ALU1, AHE3. Okay, these are some of the example of the restriction enzyme. Okay, just hold on. I need to charge the laptop. Just hold on.